Bismillahirrahmanirrahim. Assalamu alaikum. How are you? In this video, we will discuss about plasmids in details. First of all, their introduction, then their characteristic, and then their types. In very simple form, we will discuss it. Plasmids are actually small. They are circular and mostly in double bond and double-stranded DNA molecules. But they are extra chromosomal DNA molecule, not as a part of the chromosomal DNA molecule present in mostly in prokaryotes, such as in bacteria. They are present mostly in the yeast and also in some protozoan. But in exceptional case, they are present in eukaryotes and some plants. When we discuss the size of plasmid, their size ranges from 1 to 2 kb to 500 kb. In 1 to 2 kb size, it possesses 1 to 2 genes, and up to 500 kb size, many number of genes are present. Many number of genes are present in them. Now we will discuss about the plasmid in the basis of number. There are two types of plasmids on the basis of number. One is called low copy number plasmid. Copy number plasmids are also known as to them strength copy number plasmid, in which there are only present 1 to 2 plasmids in a cell. 1 to 2 plasmids in a cell. And the other types are known as high copy number or relaxed copy number. Relaxed or high copy number plasmids, in which there are present 10 to 100 copy of plasmids in a cell. Now I will discuss to you characteristic of plasmid. Generally, the plasmid are independent. They do not depend on the host machinery for their reproduction. They only use their own methods to produce its number, their numbers. Now I will discuss to you types of plasmids. There are four types of plasmids. One is called F plasmids. Second one is called R plasmid, and the third one is called cool plasmids, and the fourth one is called virulence plasmid. I will discuss it in detail. So first of all, I will discuss to you plasmids. Yes, plasmids are also known as fertility plasmid or six plasmids. It is a type of plasmids in which it has the ability to transfer the chromosomal gene from one cell to another cell Yeah, it's a transfer the chromosomal gene from donor to recipient. Okay, and this is the first ability of a plasmid that if a cell have no chromosomal gene that transfer, a plasmid have the ability to transfer the chromosomal gene from one cell to another cell. And the second ability is that it has also transferred the plasmids from in one cell in which there is no plasmid to another cell in which there is from one cell in which there is present one plasmid a plasmid to another one cell which is known as no plasmid. It means that there are two cells. First number one, and this is the second cell. A plasmid is also its ability to transfer the plasmids from this cell in which there is present the plasmid to this cell in which there is no present the plasmids. So it is the so there are two types. So there are two ability of the plasmid. First number, there is the ability to transfer the chromosomal cell from one cell to another. Yeah, its ability to transfer the chromosomal gene as from donor to recipient. And it is also the second ability is that a plasmid have the ability to transfer a plasmid from one cell in which there is present the a plasmid to another cell in which there is present no present the plasmid. Second type of the plasmid is R plasmids. So we will discuss the R plasmid in this palm that those cells which have the ability to show resistance against at least one antibiotic as well as to many antibiotics. And it means that our plasmids have the ability to show at least one antibiotic to show resistance to at least one antibiotics or to many antibiotics. And it's this is the first ability. And and second ability of R plasmid is that in those cells which have no R plasmid, their R plasmid has also the ability to transfer it from those cells which have R plasmids to another cell in which there is no R plasmids. Okay, now number third type is the cool plasmids. Cool plasmids. Those the cool plasmids are also known as carcinogen plasmids or carcinogenic vector plasmids. And this plasmid it has the ability to synthesize the chlorosin. Chlorosin are a protein. These plasmids have the ability to synthesize the chlorosin protein. But these cold plasmids have also the ability to show resistance against chlorosin, like stand and cannot produce kerosene. It also has the ability to produce cool plasma to show resistance against colocin. Colocin plasmids first their ability to produce colocin that is protein and then its ability to kill those cells kill those cells which do not produce the colocin. And the second ability of that is the because the colocin which are produced by them have the ability to kill those cells are bacteria which do not have the ability to produce glycine. And the second ability of the collagen protein is that it is also same to that of the R plasmid and the E plasmids. It will produce, it will, it will, it will have the ability to transfer the collagen protein or collagen plasmid from those cells which have the cool plasmids to another cell which do not have the cool plasmids. And number fourth is the virulence plasmids. Virulence plasmid. This is the last types of plasmids. These are large size plasmid. In this plasmids, it will show vibrancy to a host cell in which they are present. So virulent plasmids have the ability to show virulency to those cells and cause diseases on those cells on which they are present, on which they are they are present. And it is also the second ability is same to that of the previous all types. And its ability to transfer the virulent plasmid from one cell to those plasmid in which there is present, in which there is no present the virulent plasmid. So these are all about video today. Types of plasmid I will inshallah cell to cell transfer of plasmid I will discuss tomorrow inshallah. Thank you. Please subscribe, like and share this video. Okay.